Hi, welcome to the BYU-Idaho Tutoring Center videos. The topic of today's video is the rate of speciation. So today we're really going to be focusing on answering two questions. The first is how long does it take for a new species to form or a speciation event to occur? And the second is how many genes do we need to change in order to have a speciation event occur? So to our first question, we need to understand what the term punctuated equilibrium means. So punctuated equilibrium is just when we are looking in the fossil record and suddenly there's a new species that appears in geological stratum, and then it will persist for a space of time and not really change, and then it will disappear. And this is different than gradualism, in which we see over many different rock stratums slow morphological changes in a species until eventually new species forms. So if we look down here at this example, we have this one species of flower. It has a red center and a blue petals. If it was to happen by punctuated equilibrium, or the speciation was to happen that way, then when we look at one rock strata, we would all of a sudden see two very different species of the flower, and then throughout time they kind of stay the same. Whereas with gradualism, in one rock strata, we would see some slight differences between them as they slowly start to diverge. And then in another rock strata, we'd see more diverging characteristics, and then the last, we would finally get to the two different species. So that's just describing how fast it occurs. But something we need to keep in mind is that geological time is different than our time. So something that seems rapid or sudden in geological time still takes a very long time in our time scale. For example, if something, species exists for 5 million years, it's a pretty good chunk of time, and in the first 50,000 years of its life is when the most changes occurred in it morphologically, and when it really became a different species from its ancestor species. 50,000 years seems like a really long time for a speciation event, but in reality, in the entire span of its life, if the species has been around, that's only 1%. So this just really goes to show how this event from this rock strata to this rock strata, in which we suddenly see this new species appear, punctuated equilibrium, this could still be 50,000 years, because in rock strata, sediment accumulates so slowly that we really can't see, 10, 20, 100, 1,000 year differences. It's on the timeline of 50, of tens of thousands to hundreds of thousands of years. And so kind of the answer to this question is that speciation can occur pretty rapidly. We've seen it, we've done studies in which it does, and then it also can take a long time. On average, they did some studies and they found that it can be anywhere from 4,000 years to 40 million years, and on average it was about 6.5 million years for a speciation event to occur. And basically all we're doing is we disrupt gene flow, and that's what causes our speciation events to eventually happen. And so don't just think that species have a set timeline or scale in which they try to form a new species. It all just has to do with random events that disrupt our gene flow. And so overall, we come to know that it really takes a long time. I mean, 4,000 years is our kind of low end of the spectrum, but if our average is about 6.5 million years, and that goes to show us something about the origin of life and how old the Earth has to be to fit this long time in which it takes for species to form. So that's kind of the answer to our question. It depends on the situation, but generally it takes a pretty long time. Our next question is how many genes do we have to change for speciation to occur? Well, at the bare minimum, we can change a single gene, and that can cause a reproductive barrier, enough reproductive barrier that we have an entire new species. So an example of this is in this snail. One gene controls which way the snail's spiral goes. And so one goes this way and one goes that way. And if they are too different, then they aren't able to mate because of this mechanical isolation. They have their reproductive organs in different orientations, so even if they try to mate, they won't be successful. And so that's an example where one gene can result in two different species. It's not always one gene, it can be multiple genes as well. An example of that is in monkey flowers. There's two different species, and they're separated by both prezygotic and postzygotic barriers. So the prezygotic barriers have to do with pollinator choice, and gametic isolation. And the post-zygotic barriers, if they are able to form a hybrid, it usually has reduced viability and fertility and won't survive. So the major one that influences these two being different species is pollinator choice. And this is influenced by two loci in the genome of these flowers, and those loci determine the flower color. And so bumblebees tend to pollinate these pink monkey flowers, that one species, and hummingbirds tend to pollinate the orange-red version of this species. And so that different loci, depending on what's there, in this species it results in it being pink, and this species it results in it being orange-red. So this is an example in which two or more loci result in two different speciations. So the answer to this question is kind of like the answer to the other question. It really just depends on what we're talking about. It can be done by one, but it also sometimes requires many different allele changes or changes to genes in order to have speciation occur. I hope this was helpful. I hope you have a better understanding of the rate of speciation and gene changes that require speciation to occur. Thank you for watching and have a great day.